Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh dan salam sejahtera. Yang saya hormati pengerusi majlis dan juga pensyarah yang akan memberikan ucapan perdananya, Profesor Chuah Cheng Hock, dan sama sekali Datuk Cheng bekas mantan Ketua Jabatan Kimia yang tidak asing lagi, Ketua Jabatan Kimia, Profesor Sanaria, rakan-rakan Profesor di Fakulti Sains, juga tetamu-tetamu pada petang ini. Alhamdulillah kita dapat bersama pada petang ini untuk mendengar syaran perdana daripada Profesor Chuah Cheng Hock. Walaupun Dewan ini tak penuh, sebenarnya penuh satu kampus. Physically we are not that many, but virtually we are many. Because we have the facilities is live telecast, so everyone is at their respective computers watching us for this inaugural lecture. Before we would like to really welcome the guests for this evening function. Unfortunately, there are so many meetings and at similar time. The students are busy with their other activities. At the same time, we have other seminar, and we have one seminar in chemistry currently, but they are on the way to hear what Professor Chuah is going to talk today. Before Professor Chuah deliberating his lecture, may I give a small synopsis about his background. Professor Chuah is a professor at the chemistry department, University of Malaya, whereby his current research includes natural product chemistry, palm oil chemistry, and insect chemistry. He was born in Anzhi, Fujian Province, China, and migrated to Malaysia in 1958. So he received his early education in Chongkwok School, Kuala Lumpur, and completed his secondary education at TAR College. He then obtained his BSc degree in physics in 1976, graduating with second upper, and he stayed on to do his master degree in chemistry. And in 1983, then did his PhD in chemistry as well in 1988. He was initially appointed as spectroscopist in chemistry department in 1976. Then he was promoted to lecturer in 1989, then proceed to associate professor in 1998, and as professor in 2005. His current research interests focus on the extraction and characterization of natural products from plants and insects. His forte is in the application of analytical techniques such as GCMS, LCMS, and high-field NMR in structural elucidation. He has supervised both PhD and master students in various areas of natural product chemistry and palm oil chemistry. He is a team member of several research projects funded by IRPA and German Research Foundation. Amongst these are collaborative research projects with Malaysian Palm Oil Board, MPOB. Currently, he is supervising postgraduate students working under this project. So there are quite a number of projects supervised by Professor Chuah, currently done in MPOB. Professor Chuah has been very active and has contributed in many ways to the activities of professional bodies like IKM, Institute Kimia Malaysia, and MOSTA, Malaysian Oil Scientists and Technologies Association, and he's currently a council member of MOSTA and IKM. He sits on the board of trustees of Tan Sri Datuk Sri Beck Nielsen Foundation, and he has served in committee of the Board of Examiners for the Licentiate Examination, LMIC, of IKM for many years. Professor Chuah has published widely in national as well as international learned journals. He is one of the recipients of 2006 Publication Award. Over the years, he has contributed towards nation building exemplified by the vast number of successful graduates under his supervision, who are now holding top as well as responsible positions, both in the academic as well as corporate sectors. So today, Professor Chuah will talk about defense strategies of Malaysian termites. From there, you can see he came from physics background, doing chemistry, now talking near like biology. You can see how chemistry blend the two disciplines. So please welcome Professor Chuah for his inaugural lecture. Applause are yours. Okay.
I would like to extend my very warm welcome to all of you for attending this inaugural lecture. Special thanks to Yang Berbahagia Dato Muhammad Jamil for chairing the inaugural lectures, and Dato Cheng for attending my inaugural lectures, and our head of department, Professor Sanaria, for her presence. So in my lectures, my research activities basically based on the natural product chemistry, palm oil chemistry, and insect chemistry. And in the natural product chemistry, we collect a sample from the forest, like Estonia angustifolia, and we take it and dry it. After that, we do the extractions by chromatography separation, followed by spectroscopy elucidation to provide us a new dimeric alkaloids. And the output from these natural products research are some of the plants that we investigated are the research here. And one of them is the Estonia angustifolia, provide the new dimeric alkaloids. And the output from this research are published in all these ISI publications as listed here. And in addition to the publications, we produce 5 books, and 2 of the books are from the 101 plants to fight cancers and the phytochemical guides from Malaysian flora. So in the palm oil chemistry, where we have a oil palm plantation, where we get fresh fruit bunches, and from the fresh fruit bunches, we go to the palm oil mill, where we do the extractions to get your crude palm oil. And this crude palm oil can be trans-acidified to produce the biodiesel. And one of the spin-off from this biodiesel project is the productions of phytonutrients, such as the tocopherol, that is good for slow down the aging process. And the carotenoids give us a good size, and the sterol for the pharmaceutical industry, and the scoline for those energetic people. And in addition to this, we can produce specialty products like the monoglycerides as an emulsifier, and diacylglyceride as a specialty fats. Okay, so in addition to that, there are palm-based industrial solvent, additive for ultra-rose diesel, and aviation turbine fuel. These are some of the research projects in cooperation with the MPOP. So the outputs publications are listed here. Most of them published in the Journal of American Oil Chemical Society. So the next one is the insect chemistry, where we get the chemical attractants from these fruit flies. Those are Petrocera calampore and papayae, and their relations to spathiphyllums. So these are the fruit flies that are attracted to this by the chemical attractants, the pheromones. So in the output from this, we are published in the publication in ISI, in the Journal of Chemical Ecology, Biochemical Systematic and Ecology. So the next one, the topics of my inaugural lectures is the termites. They are kingdoms, consist of the queen, the king, workers, soldiers, and the winged reproductive elites. So, in fact, the termites, we always refer them as white ants. In fact, they are not whites, They are all different type of color, as we can see by these Nasutitermes matangensis, they are brown in color, and the Hospitalithermes umbrinus, they are black in color, and the Hospitalithermes rufus, they are red in color. And the Globidermis sulphurus, they are yellow in color. So, in fact, termites, they are not white ants. They're all different, provide very colorful insects. So the distributions of these insects are basically from the 40 degrees north to 40 degrees south. And in termites, there are approximately now 3,000 species worldwide. Around approximately 3,000 species of termites worldwide. So in Malaysia, there are approximately 180 species represented by 3 families: Termites, Rhinotermites, and Termitids, and 8 subfamily as listed here, and 42 genera. So for these termites, they have to defend against themselves, against the predator, especially the ants. Well, they are small, but they are powerful tools in defense against these termites. Okay, their defense strategies from the soldiers, they have a mechanical means like biting mandibles, flicking mandibles, and plucking holes. So these are the termites, Macrotermes. They have a very sharp, biting mandibles. As can show here, they are very strong biting mandibles. These are all mechanical defense. And the Procapitomys are the flicking mandibles. The mandibles are a bit twisted. Okay, so from these Termitids, these are basically most of the termites are using mechanical defense. So example by the Cryptotermis is a plucking hole. By this, the head, that is a primordic head that can plug the hole to prevent the entry for the enemy from entering the colonies. Okay, so the other one is a Clytotermis. They are the biting mandibles. So these are basically, they all are from this family. They are basically all are mechanical, utilize mechanical defense. Whereas from the Macrotermitids, another subfamily, we have Macrotermis pakistanicus and Macrotermis carboninus. These are all the biting mandibles. As shown here, they are basically mechanical. But sometimes they are giving us some of the chemicals like quinone to repel these predators. And the nets are very big. You know, the Macrotermis, the nets are very big, as high as a human being. And the moats, all these are very high, tall. And the other is the combinations of these mechanicals and chemicals. And this can be illustrated by this Procapitotermis with a twisted mandible for the flicking mechanism. And they not only give out the mechanical, sometimes they produce some of the chemicals, more to repel the assailant. And the other one is the Termis. By the Termis conis, as the twisted member also, look at that, the twisted member for the stamping actions. So in these termites, there are, other than from this subfamily, we have Amitermis that provide the chemicals. These are some of the monoterpene and the sesquiterpene that produce as a defense chemical to repel their predators. And from the Homenotermis, we have 2 species here, Homenotermis elenoma and Homenotermis for amphila. And this, they produce basically is monoterpenes. 98% are terpenoline and 68% are terpenoline from these 2 different species of Homenotermis. And the total chemical defense, mechanical, chemical, and now is the total chemical defense can be illustrated by this Longipitomys longipes that we have a pointed thing that we can shoot it out as a gun, the nasu as an extension that they can shoot out chemicals for defense against the predators. And their nets, sometimes we talk the people that walking into the forest, they stumble a black thing, then could be a Longipitomys longipes. And the other one, not only the soldiers have a defense strategy, their workers also, they have a behavior, the defense strategy as illustrated by this Globidermis sulphurus, where they have a well-fortified nest. They seal off the entry in the rapid nest repair. These are workers that they try to seal off very fast the repair of the nest to prevent the predator from attacking the nest. So this is the behavior type of work. And the recent publications are the defense mechanism of Malaysian termites that we have published it. And from there, we come to the Coptotermis. I will go to all this. Another one is the Coptotermis. The species that is Coptotermis that is shown here are the cosmic, they have biting mandibles. These are a bit nasty, the termites that destroy the house. So these are the cotylotomies destroy. They produce some of these chemical compound for the defense, and their chemicals extend to the abdomen. The storage of chemical extend to the abdomens. And from the other one, another subfamily Termitogenitorine is called Termitigidone. And from this species, they have only one species in Malaysia. Unfortunately, you cannot find in any other place. And this species, Termitogenitorine planus, only can found in Malaysia. You can see they are biting mandibles and a strong, very big heads. These are the soldiers. And from the Termitogenitorine planus, we can produce all these chemicals, all the nitro-olefin stuff chemicals that we have published in Journal of Chemical Ecology. These are some of the nitro-olefins that we get it from this. From the others, there are 4 subfamily from rhinotermitid. So another one is the rhinotermitine, where we have from the genus Rhinotermes that we produce the chemicals that is nitro-olefins. Basically, these are the nitro-olefins that can be get from this species of termites. There's Rhinotermes flavus. And from the Rhinotermes, another species that is shown here that produce the vena ketones. Now, these are all published data. Okay, so there are vena ketones. Another Rhinotermes, another species, Aquarius, also produce nitro vena ketones. Okay, so from another species, from another genus, Rhinotermes, that we have from here, Rhinotermes, that we have, there is a vena ketone also from Rhinotermes manacensis. These are a bit clearer. Sometimes we can get better pictures of the soldier. They are mechanical, and the chemicals shoot out are these chemicals as repellents against the predators. So from this family of Rhinotermes, we have 1, 2, 3, 4 subfamily. So what we have found all these chemicals. What can we utilize these chemicals? So in the previous published data on the ancestral rhinotermitates, the phylogeny of these rhinotermitates published earlier in 1975, they put their ancestor tree as this format. But this Termitogenitorine was placed under this branch. But because of our finding of these chemicals from all different species here, listed here, we withdraw the 9 to this 9 Termitogenitorine to this next branch. And we publish it, we withdraw the tree because of our findings of these vena ketones as a contact portion. So these are consistent because the development from this nitroalkane to vena ketones, and these all are linked vena ketones. So we republish it so that they have different characteristics to modify the ancestral rhinotermitids. So this was published in the Journal of Chemical Ecology. So the best is this cotylotomies. There are approximately how many species? Around 7 species that can be found in Malaysia. These are all the nasty bad termites. Their distribution are a bit narrower. The distribution in here a bit narrower in these regions. And these termites are nasty, are bad because they swarm. And they damage more home than fire and storm combined. And nationally, more than 1 in 3 houses has termite problem. That costs now 200 million property damage of these termites that swarm. And in Malaysia, about 30 million is spent each year on the termite control. And the cost of repair of termite is now 200 million annually. Damage caused by termites are nothing to laugh. They are chew. They can chew instantly at your kitchen cabinet, any wardrobe. Anything that is wood, they can take it down. Even the house, they also can take it down, you know. So, in fact, termites is the defense. The Navy are so strong, but yet so weak. When the termites come they can strike the base. Termite is so strong. The Navy is so big, but the termite strike the base. The defense, they can defend the country, but they cannot defend against the termites. So, fighting termites that's prevent methods can be used mechanical or chemicals. These are all some of the listed chemicals that we use for termite treatments. So, but these treatments, most liquid chemical treatments only have ability to suppress only. Don't document effective eliminating termites. So if your house got termites, you want to put chemicals on your house sit on the termites, on the floor of termites, maybe you may solve the problem. You only tell the termites, I chase you away. But the trouble is, chase you away, there's no problem. But your neighbor will get it. You chase her, go to a neighbor. So that is very bad, You can do use liquid treatments. So you solve a problem, but you create a problem for a neighbor. So these liquid treatments, and another thing is that you dump so many gallons of liquid onto the soil, but you are sleeping on the house that contains so much of chemicals, they leach it out. So would you like to your house sit on float on the chemicals to prevent the termites from attacking? So, but this soil termite can break down, and the effective the barrier. So we have developed a bit, a thing that can destroy the termites. So a new growth regulator that developed that can mix with the cellular foods for termite. And this compound must be what we call palatable, must be not a repellent. You don't repel the insect. The fellow must be a bit tasty also, like kacang satay lah. So the termite will eat lah. Must be a bit tasty also. So if you develop this bits, this hexafluorone, this chemical was a growth regulator. So they have the properties as a unique growth regulator. So the forest termites are not repelled, they are willing to enter termite. So you place this food, the growth regulator somewhere around here lah. So that termite will come, they pick it, forest pick up this compound in two ways through the injection, they take it and the contact with the soil as they go, they carry let them carry back to their nest lah. Okay? So this is a queen very nice queen here sitting inside. So the next step is the contaminated termites carry this growth regulator, then going inside. This transfer the active ingredient to uncontaminated termites in the exchange of food here. So this one transfer effect, it then carried through other member of the termites colony in a way at large termite, creating a colony mismanagement. And this food will feed it to the queen. Once you feed the queen good, you have solved your problem. Once the queen is killed, then you don't have a problem. So termite feed on this which contain unique regular. In time, the queen in the colony is affected and the bees and die. So this is a more efficient way of termite control or termite terminator, terminate the termites. And these are the queen. That's the more important is the queen. You can kill the soldier workers, no use. But this will reproduce very fast reproductions, very active. So it's not good. So to install another this system, you place on the ground in the termites, you drill a hole and put your instrument inside here and plug it in. First, you must monitor, you must detect whether they are there or not. When they are there, once the termite is detected, the moisture is placed with the termite bits. You put a chemical in. Once detected, you put your bits in here. Put your growth regulator, go inside. This is the insect regulator that works at stopping the insect grow. Interfere with the chitin synthesis, which they might need to form the new exoskeleton. So because of this compound that they put it in, they stop. The best thing, they have to carry to the queen. If there's no queen, then it's not possible. So they feed on this which contain the innate growth. In time, the colony will pick and die. Okay, so once you kill the queen, then you are safe. Your house will be safe. Once the termite is eliminated, the system continues to monitor for any termite. Because you kill the queen, but termite, the elites, they can fly, they can move from place to place, and your house can be infested later on, although you have solved the problem. But then it's good that you have a monitoring system to prevent them from coming again. It's a monitoring of termite, biting, eliminate, and monitoring again. So that is a system that is good for us to do. Then from this one, most of them are finished. Now we come to another subfamily, Nasutitermiti. And this one, basically, they're all chemical defense. These are all mechanical defense, and these are very nasty people. These are chemical-mechanical combinations. Okay, so now we come to the Lecystomes. There's another part of the termites that we have Lecystomes resinidae, where we collect, the nest on top, but the flower will come down on the tree, so you have to collect from the tree here. termite in the field. You collect it and you use the sampling kits, test it. Then you put it switch and you can screen on the spot screening and check what type of compound they're present from the termites. And a quick way of screening, develop a quick way of screening the termites in the forest, because we don't want to stratify the nest. Because if we find that something different, new compound, something not we haven't encountered, then we will take the whole nest to take the whole nest of the termites to do the chemical analysis. Okay, so from this Lecystomis leporata, these are the species that show some are clear nationality, some are the termites, all are different type of species from Lecystomis. From the genus Lecystomis, we have Lecystomis leporata, nationality. And we are collected from all these different parts of the Malaysian, Peninsular Malaysia. These are published data, all these are collected of different parts of Peninsular Malaysia. And in the chromatogram, we analyze chromatogram. These are all the monoterpenes, these are all the diterpenes. And from the chromatogram, you can see that there are differences between the different species. This one is another species A. So from the chromatogram, we can distinguish the species that they are all different. And to further confirm, we do these are the compounds that we show that we have found in the monoterpenes that were found from the termites, and they are diterpenes. These are all different types of diterpenes that we found of different structures that is unique to the termites. And because of they are able to distinguish in the chromatogram, then we have to do a discriminant analysis to differentiate the species. Based on the monoterpene, diterpenes, we can differentiate the species. And after differentiation of the species, you want to know what is the genetic similarity or differences. By doing the calculation, we know that the Lecystomis nationality and species A, they are closer to each other than the leporata. So these are the things that we can do discriminant analysis and genetic similarity and differences. And from the next one, we go to is the Nasutitermes. From the genus Nasutitermes, we have the Nasutitermes havilanti and Nasutitermes johoricus and Longinasus and matangasis. They are all different types of species of termites from the genus Nasutitermes. And they are collected from various parts of Peninsular Malaysia. And from the chromatogram, you can see that they are Havilanti, these are monoterpene, these are the diterpenes, and matangasis, the only two pics here, diterpene, these are the monoterpenes, johoricus, monoterpene, diterpenes. Another species is a monoterpene, diterpenes. And Longinasus, they are called, this one is a bit different. They got two types of soldiers. They got major soldier and a minor soldier. Their chromatogram quite are the same because they are from the same colony. So they must not know much variations, but there is a compositional variations. And these are the monoterpenes that was taken from the Nasutitermes and the diterpenes. All these are diterpenes that we get it from the Malaysian Nasutitermes. These are all chemical structures. Okay, and we do the same thing, the discriminant analysis to separate how these deities are different from each other based on the monoterpene, diterpene, and furthermore, we want to study their genetic similarity and differences by calculating which one is closer to each other species. Longinasus closer to species 01, and they are closer to Nasutitermes havilanti and johoricus, then followed by Nasutitermes matangasis. So from this, we come to the other one is the Hospitalitermes. Another one, these are a lot of slides showing these are Hospina favelantis. They are bringing the brown color, bicolor, two different color, and Hospitalis are the different type of color here. And Umbrinus are all black throughout. All these there are 4 species from here that we analyze, collected from different parts of Peninsular Malaysia, and we analyze by chromatogram, and we see that they are can be differentiated by color, favelantis. They are distinguishable and hospital from their chromatogram. But one thing is that from the hospital umpinus, they got even one single species. They produce 2 different types of chemotypes, A and B. They are we cannot identify. For morphologically, they are same species, but they generate 2 different types of chemistry. And these are the similar type of molotopin that is given by the hospital termites, and the titerpins that is shown here. And similarly, same techniques apply for the discriminant analysis to differentiate between the different species based on molotopin, based on titerpin, and later on, if we can differentiate, we want to how closely they are related to each other by calculating the genetic distances. Then we construct a dendrogram on the genetic distances, and the hospital umpinus B is closer to hospitalis, then it's A, then by bicolor is a bit further away. So from the number indicates there are close relationship between the different types of species that we have. And from here, we come to the next one is the Puppy-termites. There are all these Puppy-termites. From these Puppy-termites, we have Sarawakkensis, Jimenez, and Singaporeensis. They are collected different part of Malaysia. This one all different part of Malaysia that we collect it. And we get a chromogram. And the Puppy-termites, Jimenez, and Sarawakkensis, you can see all the chromogram are different. Singaporean species B, they are all different. From the and show, but we confirm by discriminant analysis. These molotopins, and this one is titerpins, and they provide some of new chemical structures, compound from this species. So discriminant analysis show that they be able to describe their separation based on molotopin, titerpin, and later you get a genetic similarity and differences to get how closely they are related to each other. Because this information is useful. Later on, when we use the information, what we study in chemistry, we apply to biology later on part. So the publications on this genus Puppy-termites is already published in December 2005. And next one is the Longipitermis longipes. Another genus that will come up from this Longipitermis. They are major soldier, minor soldiers. They are all different. And from there, we can show by the thin layer chromatography, you can see they have a difference in the chemical composition. They are all different chemotypes. And they are collected from different parts of peninsular Malaysia. And from the chromogram, we can see that the major and the minor soldier, they are quite closely related. But from one species, chemotype A, B, C, this one is more complex. They not only produce different types, two types of chemicals, but three different types of chemicals. And this chemistry is very close because Longipitermis are close by colony. They can produce one colony nearby. Produce this chemical just nearby, or the sympatric colony nearby produces different type of chemicals for same type of species, or aeropatric far away. Aeropatrics far away. They all produce different types of chemistry or different type of chemotypes. And these are the chemistry that build. And from here, we get the compound. This is longipine, the spirotetracyclic, named after Longipine longipes, bicyclic titerpins. That is a compound that we collected, illustrated from this species. And from here, A, B, C, they are differentiated based on molotopins. Based on titerpin, their discriminant analysis are separated. And how closely they are related, but major and minor, not much different. They are genetically close together. But from this one, from this one to the other B, they are close, but C is quite very far away. The chemistry is different from the chromogram. And from the analysis of amitermis, homatotermis, Puppy-termis, Havilandermis, I didn't discuss Havilandermis. These are lysotermis, leucotermis, longipes, nasutibins, saprophytermis. The analysis of the chemistry on here, what we can use to the analysis for this is that this proposed parallel typologic regressions of social medical concordant with the evolution of chemicals. These are Emerson's proposed like this. So after we have find all these chemicals, start filling up, then we find that this is not very suitable. So we have to do another way to make it as a monophyletic line, the concurrent with the evolution of chemicals. And this one we published the paper. So titerpin synthesis, Longipitermis, longipes, here, then we rearrange it. From here, all this we rearrange the reassignment. Then this is where there are regressions of mandibles. You look at the mandible very big, as you go around, become smaller and smaller, erosion of the nasut. So this is the more proper way of doing from dipyretic, monopyretic regression of social mandible concurrent with the erosions of chemicals. All the chemicals here start evolve, so that we give it a different tree. So we have to modify from the dipyretics to the monopyretic line. So these are the publications from the Longipes. Okay, so this here we have done all this basically on the biological aspect. Then we come back to the chemistry. From these 5 species of termites, 5 genus. So what are the chemistry that provide? The nasutitermes provide 2 new structures as listed here from the nasutitermes, and we publish it. Not only they provide chemistry, they provide some of the new structures that we have already published it. And from the hospititermes, we have all these are all the different structures that we have published it, some of them in the tetrahedral letters. And this one actually is Umbrinus that provides a lot of new structures. From this species, that is same as sometimes it's just lucky. You work on this species, they give you a lot of structures. And all these different structures that they come out. And these structures that we elucidated using the MMR spectroscopy, we have do a COM, carbon-13 MMR. Then we can use that to pulse it, the proton at 135 degrees to differentiate our CH2 signal. All these are CH2 signal, CH and CH3 signal here. So this depth is useful to know which are the CH2, CH3, CH signal, as is indicated here. And from here, we go to the COSI, HH-COSI, 2-dimensional MMR to link between the protons and the 2 protons connectivity. So this proton-proton connectivity, you can connect proton here nearby, but this proton-proton connectivity is transcated at a quaternary carbon. So this one is HH-COSI, then you get a HC, proton-carbon, the link of proton to the carbon skeleton of the structures. And these are very good because they provide a lot of structures for us to elucidate. And from the another structures, the same, all different type of things that we do. These are different type of structures. We imitate another structures that we use, proton-proton COSI, HT COSI for the elucidation of structures. And sometimes we need a NOE, nuclear oasis effect. These are all discovered by Noger. To 2-space proton-proton interactions. So this one you establish structures. Sometimes the structure is too big. Sometimes it's not that easy. So you need a lot of techniques to do the interpretation. This one is another one, another structures. These are some of the things that come up from the hospital Termites umbrinus. And so most of the techniques that we use, we require MMR spectroscopy. You need an inadequate experiment to establish the backbone, the carbon change of the structures. Actually, they give you inadequate, actually the adequate, very bad. They put inadequate, not enough. No, carbon-carbon connectivity, you already established a skeleton. Why they want to limit inadequate? I don't understand. So it should be adequate for you to in. Then the other one you can study is the HMPC, heteronuclear nuclear multiple bond connectivity. You can study where is the bond, the carbon to the 2J, 3J coupling. Study all these things. Okay, so the other one is proton-carbon COSI, proton-carbon linkage. So these are some of the techniques that we use in the MMR spectroscopy. But in the MMR, sometimes very dull, they sometimes discover disco experiment also have, Have you come across some they have a disco? Sometimes they make it more interesting. Ha experiment or tango, all different type of MMR, not shown here. These are the NOSI, 2-space, 1-dimensional proton-proton induction. NOSI is for 2-dimensional MMR experiment. So these are the build-up of the MMR spectroscopy that we use for the structural elucidations of the compound. So not only the structures, sometimes we need the mass spec to get the molecular ions. So in the molecular ions, they come up from here. We can get it to the fragments ion. How do we fragments ion? There are fragments. So you get a parent ion. You go to the mass spec, you get a fragments ion. Or panel iron, you go to Dr. Irons. But we cannot find any sun iron. Yeah? We only can find doctor, but we cannot find sun. No sun here. Only Dr. Irons only. So in the Termites singaporensis, they provide the new compound that is listed here. Some from the Termites singaporensis. Okay, they are the campaign skeleton that is already from the Malaysian termites. Then from the Longipithemis longipes, we got the bicyclic, and the spirotetracyclic longipine structures. These are from some of the structure that we can elucidate from these termites. Okay, from here, Termites, all these type of compound that we analyzed, that we investigated. Okay, so what can we do? We want to look at the biosynthetic pathway. How this thing is from monocyclic to bicyclic, longipes, tricyclics, and repeating, tetracyclic. So there is a thing from the development from cyclotrinoatin by this Longipithemis longipes to Ascetomis and Asutitomis. And from here, we can link how from this bicyclics to tricyclic, tetracyclics. So something that's try correlations between the biosynthetic pathways and the evolutions of these termites in their pathway. Okay, the next one is the Coptotermites. These are the nasty people that destroy the thing. What can we put it into the better use? Notorious, but they are the big eater. They eat the woods, very strong, and they work 24 hours. We work 8 hours, these work 24 hours. What can we put into better use? Better ask them, don't come and destroy all the houses. Can we put into better use? One of the better use is that we publish in the Nature, there's a point towards the bioenergy. Scientists have discovered that microbes in the guts of termites convert the food to biofuel. This one already published in somewhere Sunday Star, November 2007. So this one, if we can ask them, don't come, better put into a better use, then will be more beneficial to us. And the next generation of this will be next generation biofuel. Can they provide the energy that we require for the world? So in this, I would like to acknowledge some of the people here, and I would like to dedicate the lectures to the late Dr. Tuapong, who was inspired to this type of work. Okay, then I would like to thank you for your attention.
Okay, thank you very much, Professor Chuah, for a very enlightening lecture. I'm sure now we learn a lot about termites. There are 3,000 species and just about a few hundred in Malaysia, but we have one special ID for Malaysian termites. And we learn a lot about how strategic they are, these termites. Maybe we can apply this to our defense. Maybe we can speak to the defense department how to strategize it. And also we know from the lecture how many chemicals are there. We can extract from there, and maybe we can look, maybe we can find solution to it. We synthesize a new chemical so that this termite can confuse themselves, let them fight among themselves, at least we can solve the problems. Anyhow, this lecture, I think, really tells us how the work of Professor Chuah and the group in really identifying, and also let us know how from physicists to chemists, now he's biologist, so he's a real scientist. I think we really appreciate your lecture. I think very enlightening to us. I hope those audience in front of their respective PCs really enjoy the lecture. And shall we thank Professor Chuah in the usual way? Thank you, Prof. Chuah.